This is a channel which is going to help you to obtain a distinction in life science. Keep in touch. We'll be posting each and every section of life science until the end of the syllabus. Thank you. Let's look at DNA code of life. When we talk about DNA, we know that we have been using this in the movies. You have been seeing this in the movies about DNA. But what is DNA? If you remember in grade 10, we covered some nucleic acids whereby we talked about DNA and RNA. So in this case, we are going to look at it in detail so that we know exactly what is DNA. This is one of the topics which you can answer very well and get all the marks. It is found in paper 2. So it means that you have started your journey, you have started it with paper 2. Make sure that you understand this topic, because one topic links to another topic. If you don't understand one topic, then it means that next topic will be a challenge for you to understand. Let's start without wasting time. So you will be able to find us on YouTube, on Facebook, and our website. So you'll find the material and you'll find videos which we'll be posting. Don't forget to subscribe so that you don't miss out for this time. Let's look at DNA. Introduction about DNA. DNA is often referred to as a code of life. Why? Because it contains instructions on how to build various proteins. You know that our body is made up of proteins. Therefore, if our body is made up of proteins and what makes protein to be made is DNA, we say that DNA codes for life. So let's look at where is DNA found. If you look at this, you'll find out that DNA is found in the nucleus. This is the structure of the nucleus, if you remember in grade 10. So basically here we are looking for the nucleus. So the DNA we can ask you a question that where is DNA found? So basically here we can see that DNA is found in the nucleus. So if you look at it, we call it chromatin network. Remember, when chromatin network condenses, it forms what called the chromosomes. If you uncoil and you go into detail of the chromatin network, you'll find what called DNA. But what is DNA in full? Because you can ask yourself, you have been talking about DNA. What is it in full? We're going to see it in detail. What is DNA in full? So we say that if you magnify, you'll find out that if these chromatin network condense, they form what called chromosomes. If you look at the chromosomes, if you make it bigger, or if you try to magnify it, you find the DNA is found there. But we shall see that DNA is made up of different bases and different nucleotides. We are going to look at them in detail. Don't stress too much. You will understand it better as we continue. We have seen that DNA is found in the nucleus. So when I talk about chromatin network, I talk about chromosomes, I talk about DNA, I talk about genes. Those are the same thing, but on different scale. For example, I can say, where do you stay? You can say that I stay in Africa. Where exactly in Africa? In South Africa. Where exactly in South Africa? In Gauteng. Where exactly in Gauteng? In Johannesburg. Where exactly in Johannesburg? In Florida. Where exactly in Florida? So you talk about different scales. So when you talk about chromatin network, talk about chromosome, talk about DNA, you talk about genes, almost the same thing, but on different scale. However, when you are defining, you can define them differently. So let's look at the nucleic acids, because we're saying that this DNA is an example of a nucleic acid. So let's look at the nucleic acids. Nucleic acids, it means that the acids which are found in the nucleus. So we are saying that these nucleic acids, we're saying that consist of nucleotides. These acids, they have what called nucleotides. They are building blocks. They are building blocks, or the monomers. So sometimes they can call you, they can say the smallest unit of a nucleic acid is called what? It's a nucleotide. The smallest unit of DNA is called what? It's a nucleotide. Those are questions we can ask you in an exam. The smallest units, or what are the monomers of DNA? What are the monomers of nucleic acids? Those are the nucleotides. Don't forget that. the meaning of the nucleotide. So let's look at it. A nucleotide is made up of, number one, a phosphate group. You have to know this. A phosphate group, yes, the phosphate group is represented as a circle, and then is made up of a sugar. Basically, the sugar it depends on which type of nucleic acid. It is either deoxyribose or it's a ribose. When we talk about deoxyribose, it means that D means negative. Oxy means oxygen, ribose. It means that this kind of sugar lacks an oxygen somewhere. That's why you call deoxyribose. So, it's made up of ribose, a sugar which you call ribose. It means that this one is full. I don't need to explain that much. So it means that it is just full. Yes, you will study these sugars more when you go to university. So because this sugar has 5 sides, therefore this kind of sugar is called a pentose. Penta means 5. So it's made up of 5 sides, which means penta. Yes, so because it is a sugar, OSE indicates a sugar, like saying glucose, yes, maltose, ribose. So because this is a sugar, so it is the type of sugar, yes, is called a pentose. But what is its name? Because of 5 sides, therefore it's called a pentose. But what is its name? It is either deoxyribose or it is ribose. So in exam, what are we going to ask you? We are going to ask you, name this sugar. Don't call it a pentose. If it's DNA, tell us it is deoxyribose. If it is RNA, tell us it is ribose. So we shall see when you go to the questions very well, so that you can answer these questions without stress. Another one is called nitrogenous base. So nitrogenous base, there are 5 types. We have what called the adenine, the thymine, the guanine, the cytosine, and the uracil. If it is DNA, then it means that it's going to have some specific nitrogenous bases. If it is RNA, it's going to have some specific nitrogenous bases. So we will see which one goes to the DNA and then which one goes to RNA. So these nitrogenous bases, they look like this, or sometimes we can make it a different shape, depends on which type of nitrogenous base is that. So basically we see that the structure of nucleic acid is made up of a phosphate group, is made up of a sugar, and made up of a nitrogenous base. Which kind of sugar? It will depend on which type of nucleic acid is that, either DNA or RNA, and which kind of nitrogenous base is it having. It will also depend on which type of nucleic acid is it. Is it DNA or is RNA? So sometimes they can ask you, draw the structure of the nucleotide. So the structure of the nucleotide looks like this: a phosphate, which is represented with a P, yes, a circle, or it's a P, and then it is bonding with the sugar, which is the S. Yes, this is a phosphate sugar bond or sugar phosphate bond, and then the sugar is bonded to a nitrogenous base. So this is the structure of a nucleotide. In exam, you don't need to shade this. How do you draw it? You just draw it like this: P, S, and N, depending on which type of nitrogenous base, so that it will take the shape. However, even if you draw it as a rectangle, it still is fine. But this one must have 5 sides because it is a pentose, and this one must be a circle. So that's how you can draw it in exam. So it means that now you have marks already. You have questions you can answer. If you visit the 2020 November-December exam, you'll find out that they brought this question, whereby you need to describe this, and you'll be able to get marks here. Yes, each point you speak is a tick. Then let's look at the types of nucleic acid. As we say that there are 2 types of nucleic acids. Number 1, DNA, that is deoxyribonucleic acid. We call it deoxyribonucleic acid. What is it? Deoxyribonucleic acid. This acid found in the nucleus lacks oxygen somewhere. This acid found in the nucleus has a sugar, which is a ribo, and this sugar lacks oxygen on a certain carbon atom or somewhere. So you basically, no one is going to ask you to explain all this, but I am trying to explain to you so that you can understand what is DNA in full. Yes, so that is deoxyribonucleic acid. When you are writing what is DNA in full, you don't add SE here. No, you don't add SE because this is not a sugar. It is a molecule which contains that sugar. That's why we don't say deoxyribose. When you say deoxyribose nucleic acid, you get it wrong. It is deoxyribo. There is no SE here. Deoxyribonucleic acid. So these acids found in the nucleus, they have a sugar which lacks oxygen somewhere. That's the meaning. Then we have another type which called RNA, which is ribonucleic acid. This molecule has it is just a complete molecule. So it means that when you are writing, don't say that ribose nucleic acid because this is not a sugar. It's a molecule which contains a sugar. So we say that it's a ribonucleic acid. You remove that SE. If you say ribose nucleic acid, your answer is going to be wrong. So you're supposed to be ribonucleic acid. So you have two types of nucleic acids. Number one, you have DNA, and then number two, you have RNA. Those are the two types of nucleic acid. So we're going to look at this in detail. And this DNA is the master of all. So it controls all the activities which are happening in your body. All structures which are developing in your body are being controlled by DNA. So that's why you say that DNA codes for life. Why? Because it controls all activities. Without DNA, you are nothing. If there's no DNA, our cells are useless. So with the DNA, then we find ourselves there. And if any problem happens on DNA, it's going to affect your physical appearance. We will see this as we continue. DNA. Let's look at it. Where is it located? The types of DNA we have depend or will be determined by where these DNA are found. Number one, we have nuclear DNA. It means that it's found in the nucleus. It's called the nuclear because it's found in the nucleus. You can't call it nucleus DNA. No, when you're adding something on the nucleus, then it becomes nuclear. It must end with R. Yes. Then you have mitochondrial DNA. You don't call it mitochondria. If you're adding something on the mitochondria, it becomes mitochondrial DNA. This one is found in the mitochondria. And then you have choroplastic DNA. This is found in the chloroplast. They brought this in 2020. They brought this in 2020 exam, national exam, and people couldn't remember this. Use this material is going to help you to write correct answers, and you'll be able to write without stress. All the information in these slides is very important. Watch the video. If you don't understand a certain concept, watch these videos so that you can understand. Even if the teacher is not there or is having a problem, still you can use this material and you'll be able to pass your exam. That's why we are designing this material for you. So we have nuclear DNA found in the nucleus. You have mitochondrial DNA, which is found in the mitochondria, and then you have choroplastic DNA from the chloroplast. So if you look at it, it means that the type of DNA will depend on where they are found. So nuclear DNA and the mitochondrial DNA, they are found in both plants and animals. While choroplastic, because chloroplast, it makes the plant to look green because of its component, which is chlorophyll, then it means that choroplastic DNA is only found in plants or other green organisms. Yes. So they can ask you, name two where you can find DNA in animal cell or in animals. Therefore, the answer is going to be nucleus, and then another answer is going to be mitochondria. Don't confuse the type and the area where that type is found. Students always confuse this. Please don't confuse where DNA is found and where and the type of DNA. All right, let's continue. How is DNA discovered? How did you come up with this DNA? History of discovery of DNA molecule. So here we're going to just look at just the basics, how DNA is discovered, just the small basics of how DNA is discovered. All right. Welcome back to our class. We are going to look at the different people who were involved, the major people who were involved in the discovery of DNA. James Watson and Francis Crick, these two, they were working together to try to solve the problem of this DNA molecular structure. As they were working, also other two people were also working on the same structure, that is Maurice Wilkin and Rosalind Franklin. Before I continue, let me just be clear. James Watson is a guy, Francis Crick is a guy, Maurice Wilkin is a guy, Rosalind Franklin is a lady. By the time this DNA was being discovered, ladies were not regarded as people who can do something. So in actual sense, there were some issues of gender. So Rosalind Franklin was taken to Maurice Wilkin just to be an assistant. So Rosalind Franklin worked with Maurice Wilkin, and Rosalind Franklin discovered the structure of DNA using X-ray diffraction pictures. So without the concern or without the knowledge of Rosalind Franklin, Maurice Wilkin took the picture to James Watson. In actual sense, Maurice Wilkin used the Rosalind Franklin pictures and then took them to James Watson. Once James Watson and Francis Crick, the moment they saw the picture, then they had to figure out, or it awakened their brain, and they got an imagination how the DNA structure looked like. So they used that information to find out the further information about the structure of DNA. Unfortunately, Rosalind Franklin died before a Nobel Prize was given. But before she died, she published an article about DNA structure. The same time James Watson published the article of DNA structure. Before she died, she published an article concerning about DNA structure. Also, James Watson also did the same. But because by then people were not recognizing ladies too much, so they took James Watson's paper to be on top, and then Rosalind Franklin paper to be on bottom. And then what happened? They recognized James Watson, that is the one who published first. Therefore, the credit goes to James Watson and Francis Crick, other than Rosalind Franklin. Yet it was the hard work of Rosalind Franklin to be the person who started with the discovery of this, because James Watson used the X-ray diffraction pictures to explain more detail concerning about this. So this shows that we should not undermine ladies, that they cannot do anything. They have ability to do the work guys can do in terms of education. So we should stop gender issues. We thought that ladies cannot do anything, which is not right. Even up to now, this perception is still available that ladies cannot do anything, but this is wrong. Why? We have a lot of things which are being made by ladies, and these ladies, they are helping in our community. And so this is just a clear indication that that even ladies can do something which is important in science. So we want to see more scientists who are ladies coming out like Rosalind Franklin. So unfortunately, Rosalind Franklin died at the age of 32 before the Nobel Prize was given. So in 1962, that is James Watson, Francis Crick, and then Maurice Wilkin, they received a Nobel Prize determining the molecular structure of DNA, how DNA is a double helix with complementary base pairs. But if Rosalind Franklin was there, then it means that Rosalind Franklin would have obtained a Nobel Prize instead of Maurice Wilkin. Why? Because Rosalind Franklin did a great job than than Maurice Wilkin, because Maurice Wilkin just stored the information of Rosalind Franklin and took it to James Watson and Francis Crick. So how do we, let me just repeat. James Watson and Francis Crick, they were working together to find out the molecular structure of DNA. Rosalind Franklin and the Maurice Wilkins, they're also working together. They found out the structure of DNA using the X-ray diffraction picture. This structure was taken by Maurice Wilkins to James Watson and Francis Crick, and then was used to discover by James Watson and Francis Crick to find more that DNA is a double helix with complementary base pair. They found out that the DNA consists of adenine, thymine, guanine, and cytosine. And they found out that adenine, the percentage of adenine is equal to the percentage of thymine, and the percentage of guanine is equal to the percentage of cytosine. Therefore, they say that adenine complements with thymine and guanine complements with cytosine. Therefore, they say that these are complementary base pair. This is how this came about. This that it is a complementary base pair. This forms, because there are two strands, it forms is a double, and then these strands they twist around because it is in a wave form. Yes, it forms a helical. Because it is two strands or two helicals, then it forms what we call a double helix. And then say that DNA is double helix with a complementary base pair. That's how they came about with the structure of DNA. How do we ask in exam? Who discovered the structure of DNA? That is James Watson and Francis Crick. Who discovered the structure of DNA using the X-ray diffraction pictures? Then that is Maurice Wilkin and Rosalind Franklin. In which year was DNA discovered? That is 1962. Which one of the following obtained the Nobel Prize of the discovery of DNA? Crick. Then they can give you different options, but in this case they can. You have to know these two names because we can mix. James Franklin and then Maurice Watson. In this case, you have to know. Yes, because you see Maurice, you think that that's the person. Yet they say that it is Maurice Watson, whereby that person doesn't exist in this case. So James Watson, Francis Crick, Rosalind Franklin, and then Maurice Wilkins. Yes, it's supposed to be like that. You have to know them. Don't confuse them. Some people say that these names are complex. How many people do you know in your class? There are so many people you know, but you know them, their names. If you're a soccer supporter, how many people do you know? You know almost the whole team, but these are just the 4 people, whereby you can still memorize these people, and then you are able to get marks just in case that a question has come. So that's how we can ask questions concerning about this. After this, we will look at the questions, more questions which are regarding to this topic. Then let's look at the detailed structure of DNA. DNA profiling. What is DNA profiling? DNA profiling, when you are going for DNA testing, you are going for DNA test, we are just going to do what you call DNA profiling. So we make the profiles of DNA. So how is it identified? You use the, it is shown on the screen as bars. So if the bars are corresponding, then it means that those organisms or those individuals are related to one another. So DNA profiling is a technique used to identify someone's unknown DNA profile using a known DNA profile. For example, you are looking for your father, and then you don't know your father. There are two guys who have come, and these two guys, they are claiming that you are the son or you are the daughter of those guys, but only one guy will be the father of that daughter or that kid. So now I know the DNA profile of you, I test the DNA profile. So your DNA profile becomes the reference point. So that is the known DNA profile. So this non-DNA profile, I compare it with these two people whom I don't know their DNA profile. I go and then I test their DNA. If I find that one of them corresponds with my DNA profile, then it means that that is my father. If the two of them, no one corresponds to my DNA profile, then it means that nobody there is the father. So that's how this technique is used. Is a technique we use to identify someone's unknown DNA profile using a known DNA profile. So you are the known DNA profile, the father, the other two guys are the unknown DNA profile. So if they correspond, then we say that you are the father of that kid. So here is an example. Yes, for example, this is a scene, crime scene, someone committed a crime. Therefore, when I go to the crime, I find some material, for example, hair, blood, I collect that sample. When I collect that sample, I take for DNA testing or for DNA profiling. So I get the profile of the DNA. That's why in most cases, when there is a crime scene, we don't allow people to go there. Why? Because you might bypass there, and then when they come to collect the sample, your hair is included there. And then you're gonna be one of the suspects because your DNA, once they test there, they see that you are among the people who were there. That's why if there is a crime scene, don't tend to go there if you are not an investigative person. All right, so you're saying that the crime scene, this is the sample they picked from the crime scene. So here we have the suspect. We have three people whom we are claiming that these are the people who commit the what? The crime. So what they do is to get their DNA profiles, and then I start to correspond with what we found on the crime scene. So if I use a ruler and then I draw here, there is nothing here. I draw this corresponds, that's one. This one corresponds, that's two. There is nothing here, there's nothing. Then this one is three. This one, there's nothing corresponding. And then this one is four. So this one is gone. Then what about this? This one is corresponding. 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 This one is also corresponding. So all of them are corresponding. What about this? Shows that there is some which are corresponding and some are not corresponding. Therefore, we will conclude that the suspect number 2 is the person who did the crime. Why? Because what we collected from the crime scene and his or her DNA profile are exactly the same. Therefore, suspect 2 will be taken for accountability of this. So what are some of the uses of DNA profiling? What are some of the uses of this technique? What are some? Yes. Okay. Number one, it is personal identification. You know about the person someone is missing. So it's very easy to identify the person, the identity. Someone has burned somewhere, so it's easy to identify the identity of that person. Then number two, paternity and maternity. Maybe your mother, you don't know your mother, and there are two ladies who are claiming that you are the mother. They are the mother, so or father. we can use this technique to identify the actual parents of this. How is this one possible for mother? Maybe in the hospital, and they have swapped the kids. You know, when the kids are just born, in most cases they don't tend to resemble their parents, but as they grow on, they turn to resemble their parents. So by that time, still we can do what? We can find the parents there. Then another one is diagnosis of inherited diseases. Diagnosis of the inherited diseases. There are some diseases which can be diagnosed using DNA replication to know actually that this is the disease you have using using DNA profiling. So diagnosis of inherited disease, and sometimes we can even find a cure for those diseases. Then another one is criminal identification. Criminal identification, for example, this on the crime scene, they go take the samples which are there, and then they compare to the person they are suspecting, and then we find the person who they did the crime. Then forensics, maybe someone is burned, still maybe in an accident, you can't find anything, so we can still use DNA even if it is 20 years. As long as I get any remain of you, still we can find the DNA from there, and then we can correspond it to. Then they are saying that identifying suitable organ donor. It's not easy to find the organ donor. So you have to first see that if the DNA profile of you and the person you are giving the organ is compatible or is almost the same, or otherwise the organ will be rejected by that person's body. So what we do is to first identify to see the correlation between you and the person who is going to receive the organ. And then once these they are compatible, then they transfer the organ from one person to another. Then what are some of the disadvantages of this technique? Number one, it is expensive. Not everybody can achieve or can afford to do this. Not everybody. So it is very expensive. So it requires people who are, yes, well off. Then number two, human errors. Some people imagine someone is drunk, maybe a bear is drunk and then go back and then is trying to analyze. Definitely is going to have some errors. Even if it is not that, still when you're analyzing, you can have some, you are not 100% correct. There are some errors which are developed there. Yes, that's why we increase the sample size, and then also we repeat the experiment so that you increase the reliability, so that you increase the sample size. So it means that you have to be with a lot, and then you have to repeat this technique so that you are sure that what you got is exactly even what another person is gonna get. Then we shall say that that's maybe the human errors have been minimized. Not all hospitals have necessary requirement to carry out this DNA profiling, just like schools. But look at the schools, you'll find out that some schools, they have better equipments, they have almost everything students need to learn, but other schools, they are struggling. Whether they are government schools or where they are private schools, you find them that they have different categories or different degrees. What about now hospitals? Still, some hospitals are well off. They can have these techniques to be supported. Others, they don't. Others, they have equipments, but they don't have the chemicals they use for this to happen. So you see that some hospitals, they don't have necessary requirements to carry out this DNA profiling. So then it is possible to plant DNA at a crime scene, giving false evidence. Yes, it is possible. If someone wants to frame you, then automatically can be planted, and then it means that this is not a good idea of DNA profiling. So, innocent people or persons, DNA might be at this crime scene, at the scene or crime scene, even though they had nothing to do with the crime. That's why I say that you are just passing, and then your hair drops, and then when you're collecting the sample, then they also take your hair. Then you're innocent, but because the DNA profile is showing that it's you who was there by the time the crime was committed, then automatically you're gonna be among the people who are suspects. So and that's why we are saying that, yeah, DNA profile has some disadvantages. So let's look at RNA, ribonucleic acid. So once we talk about RNA, then it means that we are going to start what you call protein synthesis. So RNA and DNA are almost the same, but they differ in some few aspects. Why am I saying they're the same? Because both of them, they are nucleic acids. There's 3 types of RNA based on the function they do. So there are 3 types of DNA based on the location. But RNA, there are 3, but these ones, they are based on the function they are doing. So number 1, we have messenger RNA. As you hear the word, it is a messenger responsible for carrying genetic information that is transcribed from DNA. So it carries it from DNA and then takes it to cytoplasm or to the ribosome so that protein synthesis can be made. Then number 2, you have ribosomal RNA. That is, it means that it is forming the ribosome. It forms the skeletons of the ribosome. So this one forms the ribosome and produce the proteins based on the information received from the transfer RNA. So the transfer RNA, when it's coming, it comes to the ribosome. So the ribosomal RNA is just the skeleton of ribosome. Then you have the transfer RNA. This one changes, or it has what called anticodons. It code for the specific amino acid. So it transfer the amino acid from the cytoplasm and then brings it to the ribosome so that a protein is what is formed. We will see this when you go into detail of protein synthesis. So what is the role played by RNA in protein synthesis? Number 1, messenger RNA molecules carry the coding sequences for protein synthesis. Number 2, ribosomal RNA forms the core of the cell's ribosome. And then number 3, transfer RNA carry amino acids to the ribosome. That's what I've been trying to explain, that their function determine the type of RNA it is. What they do will determine what that type of RNA is. So there are 3: messenger, ribosomal, and then transfer RNA. So what is the structure of RNA? Structure of RNA is almost like DNA, but just a small difference of uracil instead of thymine, and then a single strand instead of double strand. For it is straight, it's not helical. So saying that a single-stranded molecule consists of nucleotides. We saw that it's also containing nucleotide, phosphate, sugar, and then nitrogenous base. But the phosphate is fine. Sugar is ribose instead of deoxyribose, and nitrogenous bases, it has uracil instead of thymine. Nucleotide is made up of sugar, that is the sugar. Which type of sugar is ribose, and then phosphate and nitrogenous bases. It has a phosphate nitrogenous bases, and then 4 nitrogenous bases of RNA are adenine, yes, adenine, uracil, cytosine, and guanine. So these are the 4 nitrogenous bases regarding to RNA. So this is the basic structure of RNA. So this is the messenger RNA. This is how messenger RNA looks like, and then this is the transfer RNA, how it looks like. Then a ribosome RNA, it just forms the ribosome, and basically for this level you don't need to know its structure. Yes. Let's look at protein synthesis in detail. Let's look at protein synthesis in detail. Thank you very much for coming back to our channel. Today we are going to be looking at DNA replication. DNA replication. So what is DNA replication? How does it occur? Why does it occur? Where does it occur? So that's some of the questions we ask on DNA replication. What is DNA replication? To replicate, it means that it is doubling, to double. So like making a copy. So that is the meaning of replicate. But the old and the new must be exactly the same to bring about the meaning of replication. So what is DNA replication? Is the process by which DNA makes exact copy of itself. So when you are writing a definition and you don't talk about DNA making exact copy, if the word exact is not there, then it means that you won't be able to get a tick. So it must have the meaning of, it must have the word exact. So that's why you say that the old DNA molecule must be exactly the same as the new DNA molecule. Then you're saying that when, now the question is when does it occur? When does, when in the cell cycle it takes place? When does it occur basically? Or it occurs during interphase. Yes, you can say during interphase or before cell division, because interphase occurs before the cell divides. Therefore, it is here you have generalized. If you say that cell division, you have generalized. Then interphase, you are just specific to when exactly does it occur, because we have different phases, but the first phase is interphase. So DNA replication occurs in interphase. So where, now another question is about where does it take place, or where does it occur? Then you are saying that it occurs in the nucleus. So it means that DNA replication occurs in the nucleus. When you're talking about DNA replication in this case, we're talking about DNA replication in eukaryotic cells. We're not talking about DNA replication in eukaryotic cells or in prokaryotic cells, because these don't have nucleus. Then why does it occur? Why should we have DNA replication? Sometimes we ask the significance of DNA replication. What is the significance of DNA replication? That is the same as asking you, why does it occur, or why does it take place? It takes place to double. Want to see the word double? Yes, to double. What is it doubling? The chromosome number. Don't say to increase the chromosome number. Yes, it's correct. But when you're mentioning, you want to see the word doubling, to double the chromosome number, or to double the genetic material, so that now each cell can obtain an exact number of chromosomes. So why does it occur? The answer we need is to double the chromosome number. So if you had 23, if you were haploid, then when you double, then you become diploid, so that now the cell divides. If it is case of meiosis, divides, then goes back to haploid, so that you maintain that chromosome number from generation to generation. Yes. So another one is results in the information of identical daughter cells during mitosis. Remember, cells undergoing mitosis are diploid. If they are diploid, it's supposed to double. The genetic material is supposed to double. When doubles, the cell must divide. Once it divides, then it means that each cell must have the same number of chromosomes. So it will be identical to the original cells. So it means that if I have 23 pairs in one cell, then it doubles, it's going to become 46 pairs. So when it divides by mitosis, it's going to go back to 23. So that is the meaning of to bring about to identical daughter cells during mitosis. So in meiosis, it is always haploid. Then when it duplicates, then it becomes diploid, whereby it is unreplicated. Yes, it is single-stranded, sometimes call it unreplicated, but after replication occurs, then it becomes double-stranded chromosomes. So it becomes double-stranded chromosomes. We will see this when we go to meiosis. So in exam, when they ask you what is the significance of DNA replication, they brought this question in 2020 November, December still, but in most cases, students, when you ask such questions, they always find a challenge to answer this question. Then how does it occur? In other words, the process of DNA replication. How does it occur? How does DNA replication occur, or the process of DNA replication? So you have to first be with an imagination in the head so that you can describe the process of DNA replication. You need to first know this, the imagination of the structure of DNA. Then you have to follow the diagrammatic representations, and then you form the two new DNA which are genetically identical. So using that diagrammatic representation, from there, then you can write the points which are needed. All right. DNA is, we say that is a double helix. Yes, it's a double helix. It means that is two helical, first helical, and then the second helical. So it has two strands. So since we want to carry out DNA replication, this helical must straighten. It must form two strands. Yes. So it must try to break. When it uncoils, it forms this. But I'll show you how do we write in exam. So it forms two strands. Yes, two strands. After forming two strands, now these bonds are supposed to break. So you can say it unzip, or you can say weak hydrogen bonds break. So you can use any of the two because they mean the same. So that is unwinding. It starts to form these two. So now it means that you have two strands which are open. Yes, it was like this. Then because it was coiled like this, so it uncoils, so it forms these two strands. When these two strands, if you bring it here, yes, it opens. When the weak hydrogen bonds between these bases break, then it exposes these two strands. Then if these two strands, when they uncoil like this, then the free nucleotides of DNA from the nucleoplasm will come and arrange. When they arrange, they are forming now another strand on it. This strand must be complementary to this strand. So I have the new strand and I have original strand. Yes, so it means that what is on this DNA which is forming must be exactly to what is this DNA which is also formed, must be exactly the same to this previous DNA. So basically I have three kinds of DNA here, but one is splitting to form two. So this is the which is original. So it means that it has this original strand. Still, this is original. It has this original strand, and then it has this new strand. So at the end, I'll have two new DNA. So I have two new DNA. So it's gonna form like this, whereby it has the original, the original strand here. It has the original strand here, and then it has the new strand. So then it means that you form two new DNA. How do you write in exam when you're asked to describe the process of DNA replication? DNA double helix. So you start by writing the DNA double helix. Yes, it unwinds the weak hydrogen bond. When it unwinds, it forms two strands. So now the weak hydrogen bond break. So I say that you can say weak hydrogen bond break, or you can say unzip. So when you mention one of that, you get a tick. To form now two strands, it forms two strands. Yes, because I want to form two new DNA, then each strand will act as a template for formation of a new strand. So template is like a mirror. Yes, so the old strand is acting as the object, and then the mirror you form another one, which is inverse you are seeing. So now this old strand will act as a template for formation of a new strand. Even this one will act as a template formation of the new strand. What do they use to form the new strands? They use the free floating nucleotides of DNA from the nucleoplasm. So it forms two new DNA which are genetically identical, each with one original strand and then one new strand. And then this process is controlled by enzyme. So that's basically what you are supposed to explain when you are in exam. So let's look at it. When we are writing, each different color means a marking point. So you should not forget about it. DNA double helix, we give you a tick, unwind, yes. Weak hydrogen bonds, they break, yes. Weak hydrogen bond break, we give you a tick. Or DNA double helix, unwind, and then unzips. Then you also give you a tick here and give a tick here. So any of that, you will get a tick. From to form two separate strands, you will need to see the word two, and these two strands must be separate. So you say to form two separate strands, or to form two single strands. So you get a tick for that. And then you're saying that each acting as a template. What is each? Each strand you have mentioned here is acting as a template. If you don't say each, you say two strands. The two strands formed are acting as a template. We want to see that word, either each or the two strands. We must see that. If you don't mention that, then you lose a mark for the template. Using, what do they use? Using free, want to see the word free, floating nucleotides, free floating nucleotides from the nucleoplasm. So when they use free floating nucleotides from the nucleoplasm, what happens? They form what called a complementary DNA strand is formed. As I showed you that it has the new strand. So the new strand becomes the complementary strand to the original one. So it forms the complementary strand, whereby adenine pairs with thymine and then guanine pairs with cytosine. The two new DNA molecules are formed, whereby these two new DNA are genetically identical. So it forms two new DNA, and each DNA is identical to the other. So basically, the two new DNA are exactly the same. That's why I said that DNA replication, we want to see the word to form exact copy, exact. So that if they are genetically identical in terms of genes, then it means that it won't affect the physical appearance of an individual. So the person won't keep on changing and changing and changing. So it will remain the same. That's why it is not affecting the physical appearance. Then you're saying that with one original strand and one new strand. So the DNA, the two genetically identical DNA formed, each is having one original strand, and then each is having one new strand, as I explained before. This process is controlled by enzyme. So basically, that's how you are supposed to write. Here we give you a tick, 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 give you a tick, a tick, a tick, a tick, a tick, and then a tick. Most sometimes we also give you a tick here, but sometimes we don't give, depending on the marking guideline. So that is basically the process of DNA replication, and that's how we are going to ask you. What about DNA profiling? Let's look at the detailed structure of DNA. In 2020, November, December exam, Life Science Paper 2, they brought this question. It was a 20-mark question. So describe the structure of DNA. This was out of 8 marks. So we say that we know that DNA is made up of nucleotides. Remember the structure of nucleotide? So it's made up of nucleotides. Nucleotides are the monomers, the building blocks of DNA. We see that all individuals have the same type of DNA, but this DNA is different in terms of what they code. That's why we have different characteristics. How is this possible? Yet all the DNA is made up of the same kind of nucleotide. Here is the question. How do buildings differ, yet all of them, they have the same material which build them? Yes, remember DNA is made up of nucleotides. So the arrangement of these nucleotides will bring up the different structure or the different composition of this DNA or the different genes they determine. So it's the same thing. If all buildings are made up of the same type or the same kind of material, maybe bricks, but the way how you set these bricks differ in these different buildings, and then they make them to be different. But if you break them down, you find out that they are made up of concrete or cement or mixed with water, mixed with sandy bricks, iron bars, all those things. You find them in all buildings, but the way how you arrange them make them to be different in shapes. So that's what happens in the DNA. The arrangement of these nucleotides make the DNA to be different from each other. That's why we see that all of us, we are different from each other. Even identical twins, they are different. How? You find one is tall, one is small, one is big, one is more clever, one so like that. So they are different. Even the so-called identical on my side, I don't call them identical because they are not exactly identical. They're just halfway. They are partially identical. Okay, let's continue. So each DNA is made up of nucleotide, is a tick, and then each nucleotide is now tell us what is the nucleotide made up of. Each nucleotide is made up of a phosphate group, a deoxyribose sugar, because it's DNA. If it was RNA, then we talk about ribose sugar, deoxyribose sugar. Be specific. Don't tell us that it's made up of a pentose. Is made up of a sugar. Which kind of sugar? Which kind of pentose? Made up of deoxyribose sugar and nitrogenous bases. Nitrogenous bases, yes. And the nucleotides are joined together by phosphate sugar bond to form a strand. So we have seen that it's made up of nitrogenous bases. So there are four different categories of nitrogenous bases. Mention those nitrogenous bases. There are adenine, thymine, cytosine, and guanine. So if it is RNA, then you talk about adenine, uracil, cytosine, and guanine. So these bases, they are complementary to each other. So in the case of DNA, they're complementary to each other. We saw it when we are talking about the discovery of DNA, whereby adenine goes with thymine and then cytosine goes with guanine. It doesn't matter when you say thymine goes with adenine or guanine goes with the cytosine. It's the same thing as long as you compare adenine with thymine and then cytosine with guanine. Then still you get a tick. The nitrogenous bases are linked together by weak hydrogen bonds to form two strands. So we saw that, yes, these are linked together to form two strands. So if it is a double helix, it means that with two strands. So the two strands, they coil or they twist to form a double helix. All right. They're saying that the two strands, they twist to form a double helix. So basically, that is how you are supposed to describe the structure of DNA, and then you get a mark. So here we give you a mark, here we give you a mark, here we give you a mark. A mark, each different color, you give you a mark because this one has been mentioned here. Therefore, you ignore that. This is a mark. Yeah, identifying them, they are complementary. Also give you a tick there, and then we give you a mark here, weak hydrogen bonds to form two strands. It means that DNA is double-stranded. Yes, is a mark, and then this double helix is also a mark. That's how we can mark you in exam. Make sure that you don't skip these points which are in different color, because those are the marking points we are looking for when we are marking. Sometimes people, they say that they have killed the paper. If the paper has killed them, they come out of the paper and say they have written a lot. yet there are no points. Some people, they write a lot, many pages, and then after that, they come back with the cartoon marks. Then say, why am I getting these small marks? It's not about how much you write, but it's about the quality you have written, meaning that the points you have given are the points we are looking for, the facts. We are looking at the facts we need. There are some facts we are looking for when we are marking. So if you skip those facts, even if you write a cloud of words, still you will get 0 from that. So note out when you are using our resources that these marks we are looking, these are the marks. The colored part is a mark we are looking for. If you don't have these notes, request them from that number below, then you'll be able to get the points. This is the structure we are talking about, the structure of DNA or the molecule of DNA. DNA, we said that it is a double-stranded molecule. Yes, it has a nucleotide. If you look at this point, is a nucleotide. It's a nucleotide. Each nucleotide is made up of a phosphate, sugar, and nitrogenous base. Yes, these nucleotides are joined together by a sugar-phosphate bond to form a strand. Yes, the two strands, you have 4 bases: adenine, thymine, guanine, and cytosine. Adenine goes with thymine, guanine with the cytosine. The 2 strands are linked together by the weak hydrogen bond. So the double helix, we have not shown it here. So these are the points which you need to know. Sometimes you just bring a structure, we hide this, then ask you what is this, what is this. Sometimes we hide this and ask you what is it? Because this side I have adenine, then this side I will have thymine. Sometimes we don't have anything here, but we bring this and then we put it here. So we ask you, what is this? So automatically this shape will indicate adenine. The shape which shows like this indicates the adenine. Therefore, if this shape is adenine, definitely this will be adenine, and then obviously this side will be thymine. So basically, that's how you can try to identify the structure. We will see more questions when we wrap up with this topic, so that you can know exactly how do we answer those questions, and you'll be able to get marks when you go to the paper. There is no way how you can fail if you are using these resources, because whatever we are talking about is what we said. So DNA, we said that is a double-stranded molecule with a weak hydrogen bond. Yes, and then we said that maybe characteristics of DNA still you can write them like that. It has what called a deoxyribose sugar. Yes, it has nitrogenous bases. You tell us the nitrogenous bases. The nitrogenous bases are complementary to each other. Adenine goes with thymine, guanine with cytosine, and then in the actual sense, DNA is double helix. So it's double-stranded, and then this double-stranded, it forms what you call the double helix. So if they bring questions concerning about the difference between DNA and RNA, so it means that you can use this point. It's double-stranded, then the other side automatically it's not double-stranded, it's single-stranded. And then this one is weak hydrogen bonds, because single-stranded has no weak hydrogen bonds. This one has weak hydrogen bonds, no weak hydrogen bonds. If this is deoxyribose sugar, then the other side is going to be, has no, it is deoxyribose sugar, the other side is going to be ribose sugar. Has nitrogenous bases, you tell us the nitrogenous bases. For the case of RNA, this one is going to be uracil. Then complementary bases, because the other one, this one is double-stranded. Yes, this one goes with this. Then it means that RNA has no complementary bases, unless it's complementing with DNA. So we will see that when you are trying to translate it. It is a double molecule, it's a double helix. RNA doesn't form any helix. Yes, because it's a short thing and it's a little bit straight. Yes, so basically that's the structure of DNA. What are some of the functions of DNA? What are some of the functions of DNA? So number one, sections of DNA forming genes carry hereditary information. So we see that DNA is very important in carrying the hereditary information. You look like your father because you obtain DNA from your father and your mother. You find out that the head looks like your father, the hands looks like your mother. So it means that these genes are being carried from one generation to another generation. What about when you resemble someone? You go to the village and then you find someone there, you resemble that person. Does it mean that something happened? write in the comment below and then tell us what is your view concerning about that. Then when I see many comments, I'll come back and then I'll give you the reason why you resemble someone far. Think about it. All right, DNA consists of coded information for proteins. Number one, it carries inherited information. The way how you resemble, you obtain it from your parents. Number two, it consists of coded information for protein synthesis. So most, our body is made up of proteins. The biggest percentage is proteins. So hormones, enzymes, which bring about the normal functioning of our body, so is made up of proteins. But it is DNA which codes for those proteins. We are going to look at protein synthesis when we continue. Yes. So, but it's DNA which codes for that. And I explained that in the previous lesson. So in other lessons to come, we will be explaining the protein synthesis. All right. So those are the two functions of DNA in our body. We saw that DNA has complementary base pairs. So let's look at the base calculations. In most cases, some books, they don't have this, but in most cases, we ask you this. Welcome back to our class. Yeah, today we are going to be looking at the process of protein synthesis. What is protein synthesis? So protein synthesis is divided into two words. Number one, it is the protein and the synthesis. When you talk about synthesis, it means manufacture, production of proteins. Protein synthesis is the process by which proteins are made in the cell of an organism to form enzymes, hormones, and the new structures of the cell. So your body is made up of different structures, and these different structures are made up of protein. So it means that if you don't have protein synthesis in your body, then it means that you can't grow, you can't have these hormones, you can't have these enzymes. So the marking point here in the definition is the word proteins are being made in the cell. This process is divided in two different phases. The first phase is called transcription. And when you talk about transcription, it means formation of messenger RNA. Then number two is translation. Translation is the formation of a polypeptide chain. The difference between polypeptide chain and the protein is almost the same, but it depends on how many amino acids are there. We are going to see the amino acids in detail. So how many amino acids are there? If more than 50 and above amino acids, then we call it a protein. But if it's less than 50 amino acids, we call it polypeptide chain. This diagram shows the whole phase of protein synthesis. This is the nuclear membrane. These are nuclear pores. So we have gone into the nucleus, and then you have gone into the cytoplasm. So this diagram you see, it's just showing you the nucleus and then the cytoplasm. We know very well that the DNA is found in the nucleus. And then this DNA, because it is a king, is the one which codes for the protein, doesn't move out of the nucleus. It's like a king who doesn't move out of the palace. He has the servants, and the servants, they go take information, what the king has said. So these messengers, the transfers, they act as servants for DNA, because DNA wants to produce a certain protein. So it will send a messenger, and then the messenger takes the information to the cytoplasm, then the transfer RNA will bring the amino acids. So when they bring a diagram like this, you have to know how to label. That's the first point you need to know so that you can pass this question. The first thing is you have to know that this is DNA. When it is coiled, if you see something coiled, is DNA. If it is too coiled, then we call it chromatin network. Then the DNA is done which unwinds, it forms the messenger RNA, and then the process of formation of messenger is what we call transcription. Then this messenger RNA will move out of the nucleus via the nuclear pore, and then it goes to the ribosome. You see, these are the ribosomes. This messenger will align itself on the ribosome, and then the
transferrin A will bring the amino acids to this ribosome, and then a chain of protein, a chain of amino acid is formed. Therefore, this process whereby the messenger brings in and then the transferrin A brings in the amino acid also, the chain is formed, is called translation. So the major difference between transcription and translation: transcription occurs in the nucleus, while translation occurs in the cytoplasm. So you have to know at least the basics of this process. Before we go to the process of transcription, so this is a simple diagrammatic representation of the whole process of protein synthesis. So you expect DNA to unwind, and then when it unwinds, it forms, it exposes two strands, but one strand will act as a template for formation of a new strand, and then this new strand is what we call the messenger RNA. This messenger RNA, we saw that it moves out of the nucleus via the nuclear pore, and then it goes to the ribosome. The moment it reaches the ribosome, then it starts the process of translation. Then amino acids are joined together, are brought by the transferrin A. They are joined together to form a polypeptide chain. So if the number of amino acids become a lot, the more than 50 and above, then it forms what you call a protein. So the protein, it must have it's a heavy molecule compared to a polypeptide chain. So now let's look at what happens in transcription. We see in the exam, what do you write when we find such questions, and then what happens in translation. Transcription. Don't forget, this is a recall that transcription occurs in the nucleus. You have the DNA, and then you have the DNA to unwind, and then messenger RNA is formed. Then messenger RNA moves out of the nucleus by the nuclear pore, and then it goes to the cytoplasm. Basically, that's what you're going to be explaining. So the first point, what you see in different color, it means a marking point. DNA double helix. You have to tell us the natural shape of DNA. DNA double helix unwinds. So that's the first step. Number two, it will say that when the weak hydrogen bonds break. Here, when you say weak hydrogen bonds break, or you say DNA double helix unwind, then it unzips. It's the same as saying weak hydrogen bonds break. What's the meaning? If you look at your trouser, you try to open it, you zip down. When you zip down, you are breaking the bond which we are holding the zip together. You understand? So when you unzip, it means that you are breaking down the bonds which are there. That's why we say that weak hydrogen bond break. Or you can say DNA double helix unwind, then unzips. So you get the same mark. One strand will act as a template. There is a major difference between DNA replication and transcription. Here, when you're talking about transcription, one strand is the one which acts as a template, while the other side, the two strands will act as a template. So something which look like a mirror, so that there is one strand, so that the new strand is being formed. Then to form the messenger RNA, one strand will act as a template, as you see it here, and then messenger RNA is formed. It uses free-floating nucleotides. When you say this, you are supposed to specify, because when you say free-floating nucleotides, in the nucleus, there is free-floating nucleotides of DNA and free-floating nucleotides of messenger RNA. So you have to be specific when you come to this. You have to say that free messenger RNA nucleotides. Yes, so these free messenger RNA nucleotides which are floating in the nucleoplasm are the one which we use. Yes, so if you just say using nucleotides, we will cross it, because we have different kinds of nucleotides in the nucleoplasm. So you have to be specific that free messenger RNA nucleotides. Then the nucleotides, the messenger RNA is complementary to DNA. So it means that where there is A, you're going to have U, and where there is C, you're going to have G. Remember, the messenger RNA does not have thymine. It has what you call uracil. That's why we say A goes with U and then C goes with G. Then the messenger RNA now has coded message of protein synthesis. It means that now it is ready to move out of the nucleus and then go to the cytoplasm, so that it can take the information DNA wants the protein to be manufactured. Yeah, that's why you're saying that now the messenger has the coded information for protein synthesis. Then we are saying that the messenger RNA moves from the nucleus through the nuclear pore and then goes where? To the cytoplasm. Where exactly in the cytoplasm? That is on the ribosome. So it basically, this is the general process of protein synthesis, basically that is transcription. So what you see in different color, it means the marking point. DNA double helix unwind, weak hydrogen bonds break, and then one strand will act as a template, and then for formation of messenger using free messenger RNA nucleotide in the nucleoplasm, and then the messenger RNA is complementary to DNA, whereby A goes with U and then C goes with G. And this messenger which is formed, it has a coded information for protein synthesis, and then this messenger moves out of the nucleus via the nuclear pore to the ribosome. Basically, that's what we want. 6 marks from here. In most cases, you see it as a short question which requires you to write these statements the way you see it. Please don't change. If you change, make sure that the different colors don't change, because those are the marking points we are looking for. And then let's look at the process of translation. Here, let's look at the process of translation. Welcome back to our class. Yeah, today we are going to be looking at the process of translation. Here, let's look at the process of translation. Basically here, now the messenger has moved out of the nucleus. It has gone to the cytoplasm, and then it attaches itself on the ribosome. So now we want to bring the amino acid to the ribosome so that a chain is formed. Number one, what you say is each transfer RNA carries a specific amino acid. It means that it has 3 bases. These 3 bases, they code for a specific amino acid. These bases are called anticodons. When the anticodons on the transfer RNA, yes, those 3 bases happens, matches the codons on the messenger. So the 3 bases on DNA, we call them codes. The 3 bases on the messenger, we call them codons. The 3 bases on the transfer, we call them anticodons. So the DNA code for the codons on the messenger, and the messenger codes for the 3 bases on the transfer, which are the anticodons. And then the anticodons, they code for the specific amino acid. That's why we're saying that each transfer RNA code for a specific amino acid. It's like a key. The other one will not enter or to open. When the anticodon on the transfer RNA matches the codons on the messenger, so what happens? This transfer RNA will bring the required amino acid to the ribosome. So it will bring the required amino acid to the ribosome, and then the amino acids become attached by a peptide bond. We call it a peptide bond, not polypeptide bond. Don't say polypeptide bond, we will cross it. It's called a peptide bond. If there are many, we call them peptide bonds, not polypeptide. When you say that, cross it. So amino acids become attached by peptide bonds, and then to form the required proteins. That's what we want. So this is what exactly what is happening. This is exactly what is happening. Each transfer RNA carries a specific amino acid. For example, it carries a specific amino acid, a specific amino acid. This cannot carry another acid, no. And then you're saying that when the anticodon, when these 3 anticodon, yes, match with the codon, yes, match with these codons. For example, is T goes with A, T goes with A, and then A goes with U. When they match like that, yes, now what happens? This transfer RNA will bring this amino acid, which these 3 bases, yeah, which code the anticodon, will match this. So it brings this required amino acid to it brings the required amino acid to the ribosome. So now the amino acids are joined together. So it keeps on bringing, and then the amino acids are joined together by these bonds, which you see, these bonds are called the peptide bonds to form a required protein, and then you form this polypeptide chain. But now it grows. Once it grows, it is more than 50 amino acids, then we call it a protein. So these are amino acid. If they ask you what is this, it is called a polypeptide chain. Yes. And then this is the messenger, the 3 bases on each, we call it the codons. And then on the DNA, we call it the codes. And then on transfer, we call it anticodons. And then this molecule where transfer, where messenger lines is up, is called a ribosome. This is messenger, this is messenger still, this is transferrin A, this is a polypeptide chain, this is amino acid. And then this is a transferrin A, it is a T, that's why it's called T. And then this is 3 bases, which we call the anticodons. 3 bases on this messenger, we call them codons. And then 3 bases on DNA, we call them codes. Yeah, thank you very much.
Others for our daily new videos.
Yeah, welcome back, guys. Welcome back to our class. We are looking for distinctions and making sure that we pass. All of us, we must pass. We must go to the university. Yeah, I've got the vote. Yes, today we are going to be looking at what you call meiosis. I know meiosis is one of the difficult topics, very simple to take, to grasp, but try to answer your questions. Let's look at meiosis. Meiosis, we said that meiosis, humans reproduce, other organisms reproduce. They multiply. What about cells? Cells, they have 2 kinds of reproduction they undergo. They have what called mitosis and you have what called meiosis. Mitosis can occur in other body cells, but meiosis occurs only in the reproductive cells. Where did you come from? Actually, meiosis was the basis of you living. Without meiosis, you wouldn't have existed. So let's look at meiosis in detail. So it's a type of cell division that results in 4 daughter cells, each will have number of chromosomes to that of the parent. So it means that meiosis starts with the one cell. This cell divides and then you form 4 daughter cells. These cells, they have the chromosome number to that of a parent. If you have started with the 4 chromosomes, it means that these cells, they will result in 2 chromosomes. That's why you're saying that half the chromosome number of that of the parent. If the man, the body cell is having 46 chromosomes, then the reproductive cells or the daughter cells which are resulting from that are going to be having 23. And then from the mother also 46, they are going to result into 23. So the process of reducing these cells into half the chromosome number, that process or that reproductive process is what we call meiosis. So what does meiosis result in? So this meiosis will result in the production of gametes. Gametes, these are the sperms and the ova, and when you're talking about the humans, and then plants, you're talking about spores, the pollen grains, you can talk about the ovules. Those are some of the things which are as a result of meiosis. So in humans or in animals, it occurs in the ovary. See this colored word, in the ovary. Basically, ovary are found in female, and then these ovary, they produce what you call the ova. So the reproductive cells in the females, we call them ova. If it's one, it's called the ovum. If there are many, we call them ova. And then in male, we call it's occurring in testes. And then in testes, what we call them spermatozoa. The cells which are being produced, we call them spermatozoa. So in female, we call them ovary. Those are the ova. ovaries. The ovaries produce the ova, and then in the testes, they produce what called the spermatozoa. What about in plants? In plants occurs in the ovary still, but what it produces, ovary produce what called the ovules. Meiosis occurs in the anthers, and then what it is being produced is what called the pollen grain. So the question can come and say that other than the ovary in animals, which other place does meiosis occur? That is testes. And what does it produce? It produces what we call the spermatozoa. And then in plants, it occurs in the ovary. It produces what you call the ovules. Basically, that's what you needed to know about this first introduction of meiosis. You know, meiosis is basically the cell divides, and the most important thing to divide is the nucleus. So we need to know the nucleus to do a recall about the nucleus, because we saw the nucleus at the beginning of DNA and also saw the nucleus in the grade 10. So let's look at the nucleus, the centrosome, and then the cytoplasm. Nucleus contain the genetic material. What about cyto means a cell, and then plasm means a fluid, the fluid of the cell. So basically, this is what you are going to look at. Let's look at them in detail. So this is a cell. It has the nucleus. Outside, you have what called the cytoplasm. Then in the cytoplasm, you have what called the centrosome. Centrosome, why do we talk about the nucleus, centrosome, and the cytoplasm? Because the DNA, you'll find it there, and then it is the centrosome which produces the centrioles, and the centrioles produce spindle fibers which hold the chromosomes in position. That's why you talk about it. And then there is a time when this one disappears, and then you remain the content of the cytoplasm. So the nucleus disappear, and then remain only with the content of the cytoplasm. So basically, that is a nucleus, and then when it brings it in exam, it looks like this. So this is a nucleus. It's a nucleus, but the nucleus has a dense body which we call the nucleolus. Then outside the nucleus, you have the nuclear membrane which has the extension which we call the endoplasmic reticulum. But because it has the ribosome, they look rough. Then we call them the rough endoplasmic reticulum. The rough endoplasmic reticulum. So basically, that's it. It has the nuclear pore, the holes where substances go through, most especially messenger RNA in this case, and other substances. Then you have what you call the nucleoplasm, the plasma of the nucleus, meaning that the fluid of the nucleus. The nuclear envelope, that's what you call the nuclear membrane. Then the ribosome, we say that the ribosome, the sites for protein synthesis. Chromatin, this one is the one which condenses to form chromosomes. And then you have the nucleolus. Then you have the rough endoplasmic reticulum. So we have explained all that. So when you look at the nucleus, or when you look at the DNA, basically that's what you see. You have, when the chromatin network condenses, you form what you call the chromosomes. So the chromosomes, they condense. You see, they have condensed, and then after that, you form the real structure of a chromosome. It looks like this. So that's the structure of a chromosome. Now let's look at the chromosome in detail. So we have seen where the chromosome come from. It cannot exist without DNA, with its specific genes. You see, it has specific genes. So let's look at the structure of a chromosome. Why do we need to look at it? Chromosome? You resemble your father because of the chromosome, because of the genes you obtained from your parents. Your father, your mother. What about those who don't resemble the father and the mother, but they resemble their grannies? We shall see. Another question, what about why? So in this topic, we're going to see why. Why do you resemble your neighbor? Why do you resemble when you go somewhere? When you go somewhere, someone can say that, hey, when you resemble someone. Why do you resemble someone? Those are some of the questions we are going to answer. And this is the reason why we need the structure of the chromosome. So the structure of the chromosome. Chromosomes are thread-like structures present in the nucleus. They're thread-like. It means that they are like strings, thread-like structures found in the nucleus. That's why we talked about the nucleus, which carry genetic material. That's why you resemble your father. That's why you resemble your mother. which carry genetic material from one generation to the next generation. So it means that your kids will try to resemble you. Yes. Then you're saying that each chromosome is made up of the DNA. We saw this, that DNA genes are almost the same thing, but on a different scale. So these DNA or these genes are tightly coiled many times around a protein called histones. So the histones, we saw this when we are talking about protein synthesis, and also when you're talking about the structure of the DNA. Yes, so they coil around to form what called the histones that support the structure of the DNA. Because the genes cannot just exist there in air, they need to have a support. So the support we call them histones. So these are the support which make the DNA strand to stand or to be where it is, and so that the chromosome can be formed when this DNA condense. All right, this is the structure of chromosome. Before, remember, we talked about during DNA replication that when does it occur? We say that it occurs before the cell divides or before meiosis. So before the cell divide, the chromosome will look like this. Yes, they will look like this. Therefore, we call this one single-stranded chromosome. Chromosome 1, chromosome 2. Sometimes we call them unreplicated chromosomes. They have not yet done DNA replication. But the moment DNA replication occurs, this chromosome is going to change from this format to this format. That's why we say that it doubles the chromosome number. Don't say that it increases the chromosome number. It increases the chromosome number. It increases the genetic material. From what to what? The best answer is double the chromosome number, doubles the chromosome number, so that when it was like this, then it becomes like that. So this is the centromere, the center of the chromosome, we call it centromere. The two chromosomes which look alike, it means that they share the same structure, yes, and the position of the genes on them, then we call them homologous chromosomes. Homologous chromosomes. So one chromatid from one chromosome and another chromatid from another chromosome, because they are homologous chromosomes. Therefore, because we are talking about chromatids of homologous chromosomes, then they are going to become homologous chromatids. Why? Because we say that they are coming from homologous chromosomes, but each part is a chromatid. Therefore, they become homologous chromatids. So so sometimes, as I say, that this is called unreplicated chromosomes, and then we call these ones the replicated chromosomes, or what you call homologous chromosomes. So each of these has two names: unreplicated chromosomes or single-stranded chromosomes, and then these ones double-stranded chromosomes or replicated chromosomes. So the number of chromosomes in each cell is a characteristic of organisms. For example, humans, we have 46 chromosomes. You'll find out that Drosophila melanogaster has 8 chromosomes. You'll find out that the sheep has over 60 chromosomes. So each organism has a specific number of chromosomes it has. And you might find out that the biggest organism has fewer number of chromosomes. So that's how life, or that's how God is. So it doesn't matter whether you are big or small, but you might have your own uniqueness. We are saying that chromosomes which are single thread or strand become double when the chromatid joined by centromere. So the center of the chromosome, we call it centromere, as a result of DNA replication. So if DNA replication occurs, as we said, now the single-stranded chromosome are going to become double-stranded. So that's why you call it unreplicated chromosome or single-stranded chromosome. The moment DNA replication occurs, it becomes double-stranded chromosome or replicated chromosomes. Yeah. So what are some of the difference between the haploid and diploid? Di means two, ploide means you are describing a body. Yeah, so half means it is derived from the word half. half the chromosome number. If a human being is supposed to have 46 chromosomes, so when you are haploid or when the structure is haploid, it has the half the chromosome number of that organism. For example, if an organism has 20, the haploid state of it is going to be 10. If the organism is having 32, the haploid number of that organism or haploid state of that organism is going to be 16. So haploid means half the chromosome number, diploid means doubling, doubling. What does it mean? Where is the double there? It means that half from the father, half from the mother, and then they double it. When they double, then they form what called the diploid or diploid, the way you pronounce it. So haploid is the quantity of the cell or organism having a single set of chromosomes. They have what called a single set of chromosomes. So if you have chromosome number from 1 to 2, from chromosome number from 1 to 10, these chromosome numbers, these chromosomes must be in pairs. So if you are haploid, chromosome number 1 to 10, they will be in single. They will be 1, 1, 1, 1. They won't be in pairs. So that's what you call a haploid. But if it is diploid, is when the cell or organism has a paired or two sets of chromosomes, paired or two sets of chromosomes, one from each parent. It means that one from the father, one from the mother. When they combine, because there are two organisms contributing this, then you form the word di, which is 2. And then ploidy means a body. Yes, it's a state which you describes organism with two sets of chromosomes, one from the father and one from the mother. That's the meaning. So for sexual reproduction to take place, sexual, it means that it must include the gametes. It must include the gametes and it must include the sex. Reproduction to take place, yes, what happens? A haploid male gamete, that is, if we are talking about humans, we're talking about sperms, will fuse with the haploid. It means that it will refuse. When you say a haploid male gamete, half of the chromosome number from the male, the gamete will have half of the chromosome number from the male. If the male is 46, the gamete is going to have, or the sperm is going to have 23 chromosomes. Will fuse with the haploid number from the female. Also now the female is going to produce 23. When they fuse together, you form fertilization. So when they fuse together, that process is called fertilization. Now you form what you call a diploid, because half from the father, half from the mother, then because it's half from different people, then it becomes di, becomes 2. They fuse together. And then when we're talking about fusion or fertilization, we are not talking about sex, because the fusion or fertilization can take place without sex. Meaning that meaning maybe they used a male just give out the sperms. So when you're talking about fertilization, we're talking about sex has already occurred, and then the sperms, they swim along the female reproductive system, and then fusion will take place. This fusion can even take place. Someone had sex at night, and then you're walking on the street, fusion is taking place. Imagine, did you know when was your fertilization took place? You don't know. Even your parent doesn't know. Okay, those are the wonders of the science. All right. If you need more questions concerning about this reproduction, go and visit our YouTube channel, which you call Bright Doctor. Bright Doctor. I'll give you the link in the description below so that you can understand it better. All right. The result is a diploid zygote. The result is a diploid zygote. So when you have a haploid, you form a diploid or a diploid zygote. So it means that when the sperm fuses with the ovum, you form what you call a zygote. But this zygote has a diploid number of chromosomes. This body has a diploid number of chromosomes. It means half from the father, half from the mother. All right, haploid, half, which is indicated, haploid is indicated as 1N, and then diploid is also indicated as haploid from the mother, 1N. For example, haploid from the father, for example, is also 1N. So when you combine them, you form the diploid, which is whole. Yes. It's the whole. Now half and half, you get whole. Yes, basically half from the father, half from the mother, from the diploid. Di means two, meaning that contribution from one person and then contribution from another person. So because this resultant body is having two contributions, then we call it a diploid. So it is indicated as 2N. We shall be using this language. So you have to know when someone uses this language, you must know what it means. So a sperm, ovum, and then you form a zygote. Zygote is where a fetus develops from. We shall see in the videos we're gonna be releasing soon. How is the zygote form? You see the sperm swimming after sexual intercourse. You see the sperm swimming, going to the ovum, and fertilization takes place. For more detailed about this, visit what you call the Bright Doctor. Bright Doctor, just a link below. Drop your question, then we shall give you the answers on that YouTube channel. But for purpose of passing exams, we don't go far away from this. But there we can describe as much detailed as you want. So sexual cells is either sperms and ova are haploid. So we call them haploid. They're 1N, 23 chromosomes in humans. So it means that the other organisms have different chromosomes. So we have what called the somatic cells. So it means that we have two types of cells. We have what called the sex cells. Those are the gametes. I say that meiosis can only take place in these cells. We don't find meiosis in any other cells, only in the gametes. So you have the other body cells. Soma comes from the word soma, which means a body. Yes. So these are body cells are diploid, which means that they are 2N. If they are 46, it means that the haploid is going to be 23. But the question when it comes, it does not bring only human. You can only talk about 46 if they have specified in the question that this is a human cell, because they can bring a cell with 10 chromosomes. They can bring a cell with 4 chromosomes. If they don't specify that this is a human, please don't use 46, because the question is not asking about humans. Then you have the 23 pair of chromosomes that result in the zygote are divided as follows. How? Number 22 pairs. These are the number of chromosomes we have in the body. 22 pairs. That is from pair number 1 to pair number 22. We call them autosomes. Autosomes. It means that how many chromosomes do we have? 44, because this is a pair number 1 up to pair number 22. We call these ones autosomes. One pair of sex chromosome, that one pair, that is the 23rd pair, we call it sex chromosome, or scientifically we call them gonosomes. Scientifically we call it gonosomes. So this pair is divided into two. We have what called the XX, which is female, and then we have XY, which is male. So it means that females always, they will have XX, and males will always have XY. There is a question which is supposed to be posed here. Why, if females are XX and the males are XY, it means that it's the males to determine the sex, because female have X, also male has X, but female don't have Y, but male have Y. So it's the males to determine the sex of the human being, the sex of an individual. So males, you must stop saying that ladies are only giving you females, they're only giving you boys, they're only giving you girls. Say, I'm giving myself boys, I'm giving myself girls. It's you who is determining this. However, there are some other factors which also determine. It can be on the side of the female. For more details, visit the bright doctor. Drop your question there. We will be able to answer those questions. If you drop the question there, then I'll be able to answer those questions. Humans have what we call the chromosomes. We say that from chromosome number 1 to chromosome number 22. These chromosomes are arranged in a specific order and they appear in a specific way. They are in pairs, and then they describe different characteristics in the organism. So let's look at the karyotype, the composition of the nucleus. Which kind of nucleus? Which kind of nucleus do you have? The chromosomes, the type. Karyo is derived from the nucleus and the type. So which type of the nucleus do humans have? That's derive the name of karyotype. Humans cells are made up of 46 chromosomes. It means that our nucleus have 46 chromosomes. And then these 46 chromosomes are arranged in pairs. These chromosomes are arranged in pairs. It means that they cannot exist as singles. What does it mean if you're not married? All right. It means that they are arranged in pairs. Each pair is different from each other. Oh, each pair is different from other. So these pairs are different. By how do they differ? How do they differ? They differ by shape, size, and genetic composition. So pair number 1 is totally different from pair number 2 by size, shape, and the genetic composition. It means that what pair number 1 determines is not the same thing what pair number 2 determines. For example, you'll find out that chromosome number 23, pair number 23 will determine the sex. You cannot find this in other chromosomes. So they vary in shape, size, and the genetic composition. From pair number 1 to pair number 22, number 1 to 22 are called autosomes. What about 23rd pair is called gonosomes, which has XX for female and the XY for male. X, you have to know this, X is longer and bigger over Y. Why? X is longer and bigger over Y. Question, why? Why is X longer and bigger? Bright doctor will answer that. But on the purpose of this, X carries more genes compared to Y. So a karyotype, an example looks like this. You see that chromosome number, pair number 1, number 2, number 3, number 4, they vary in appearance, shape, and also they vary in the genetic composition. So from 1 up to 22, we call them all this, we call them gonosomes. We call them autosomes. And then the 22nd pair, we call it gonosomes. So basically, that's it. Revision of the process of mitosis. It's advisable before you start meiosis to do a revision concerning about the process of mitosis. So guys, our next video, as we say that we're gonna be having prizes, airtime, meals. Yes, we send you a voucher and then you buy whatever you want to buy. It can be a school bag, t-shirts. We have a lot of gifts to give out on our YouTube channel. So please don't forget to subscribe. Don't forget to subscribe, like, share, so that others also benefit from this content. And then we're gonna be having all the subjects. So we are trying to increase this. So mitosis occurs in every living organism. In the somatic body cell to produce, those are the body cells, other cells other than sperm and ovum, to produce genetically identical cells. So you can have a hole on the mouth, on the cheek, and then you need meat. So what they can do, they can cut the bum and then they put on the mouth. So you move with the bum on your face. Basically, why do they do that? Because these cells, they undergo what you call mitosis. They are genetically identical. The meat from your bums is exactly the same as the meat from your face in terms of the genetic composition. So two identical cells are reproduced, two identical cells are produced with identical chromosome number equal to the original cell. It means that the parent and the babies or the daughter cells, they're exactly the same. There is no difference. That's why when you get a wound, another meat develops. Those cells which develop, they're exactly the same. Mitosis has only one cell cycle, i.e., it doesn't have that mitosis 1 and mitosis 2. No, it only has one cell cycle. Only one cell cycle. It means that meiosis, when on the side of meiosis, it has two cell cycles, meaning that it has meiosis 1 and also has meiosis 2. So guys, let's look at the process of meiosis. Yeah, the process of meiosis was a very good introduction of meiosis. Now let's look at meiosis in detail. And this will be our next topic tomorrow. Don't miss it.